Hi, I am Dr. Arsalan and today we are going to solve the MCQs on enzymes. First question is chemical which blocks the enzymatic activity at its active site. The phenomena is called. Given options are allosteric inhibition, feedback inhibition, competitive inhibition or non-competitive. So this question basically pertains to the inhibitors. Inhibitors are those chemicals which bind to the enzyme at the active site or allosteric site and inhibit or stop the activity of the enzymes. So the inhibitors which bind to the active site are called competitive inhibitors, and the inhibitors which bind to the non-active site or the site other than active site like allosteric site, these inhibitors are called non-competitive inhibitors. It's asking about the inhibitor which blocks the active site of the enzyme. So this is competitive inhibitor. Next question is the proenzyme or precursor of enzyme is called, given options are zymogen, granzyme, ribozyme or apoenzyme. Basically the enzymes contained in the body are in inactive form are called proenzymes, and at the time of need these enzymes are converted into active states. Because these enzymes are present in active state or functional state, so these may damage the body's own body tissues or organs. So these enzymes are called coenzymes or precursor of enzyme, and the technical name for this is the zymogens. So zymogens are those kinds of enzymes which are present in the inactive form. For example, if we say the pepsinogen, it is present in stomach in inactive form. When HCl is secreted, this pepsinogen is converted into pepsine. So in the absence of food materials in the stomach, this pepsine enzyme would eat away of its own stomach wall. So in order to prevent the digestion or damage to the own stomach wall, this is present in the form of pepsinogen in inactive form. The other options, granzyme and ribozymes, are the types of enzymes, and apoenzyme is the enzyme without cofactor. Next question is enzymes are chemically composed of. Given options are proteins, nucleic acids, coenzymes, are both A and B. So this is very simple question. We all know that enzymes are all proteins in nature. But here you shouldn't make hurry to mark this option proteins, because there is an enzyme, the ribozyme, which is made up of ribonucleic acids, RNA, and RNA is a nucleic acid. So all of the enzymes are made up of proteins except the ribozyme, which is made up of nucleic acids. So here we should see the option, the proteins, nucleic acid, and the option D is revealing that both option A and B are correct. So proteins and nucleic acid both are correct. So I'll tick on the option D, both A and B are correct. Fourth question is first discovered enzyme by AIIM. AIIM was a scientist who discovered the first enzyme, diastase. Diastase was the first enzyme discovered, and it converts the starch into glucose. So starch is converted into glucose by the enzyme diastase. It was the very first enzyme which was discovered in the field of biochemistry. Question is the first isolated and purified enzyme was. Given options are urease, diastase, lipase or ribozymes. As we have seen in the previous question that the first discovered enzyme was diastase, but here it should not be confused with the sentence that the first isolated and purified enzyme. The firstly discovered enzyme was diastase, but the purified and isolated enzyme, it was the urease. So urease was the first enzyme which was isolated, purified and reutilized in the biochemistry. Next question is enzymes are usually named as per their dash. Given options are origin, discovery, substrate or host. This is a simple question because most often we see the enzymes lipase because they have the substrate lipid, the protease which digest the proteins, the urease which break down the urea, ATP synthase which produces the ATPs. So most oftenly the enzymes are named after their substrate. So option C is correct, and substrate is the molecule on which the enzymes act and convert it into product. These molecules are called as substrates. Some of enzymes are also based upon their substrate upon enzymes act. These enzymes involved in the feedback inhibition are called. Given options are apoenzyme, holoenzyme, coenzyme or allosteric enzyme. Feedback inhibition is a separate process, and most oftenly the body enzymes are controlled by their feedback inhibition. Feedback inhibition means when the enzymes are converting the substrate into product molecules, and these products again control the enzyme activity by binding to the allosteric side of the enzymes. So this is called feedback inhibition. Here the first option, apoenzyme, this is the enzyme without cofactor. The holoenzyme enzyme along with the cofactor. Coenzyme is the organic component of the cofactor which is required for the enzyme activity. And allosteric enzymes. So allosteric enzymes are those kind of enzymes in which inhibitors or their own products are attached to their allosteric site and stop activity of these enzymes. So this is called the allosteric enzyme. Option D is correct, that the enzymes which are involved in the feedback inhibition are also called as allosteric enzymes. Next question is urease enzyme converts urea into ammonia. This enzyme follows dash model. Given options are lock and key model, induced fit model, unit membrane or None of these. Know that there are two kinds of models which state the function or structure of the enzymes are the enzyme catalysis. These are lock and key model proposed by Emil Fischer and induced fit model by Kochland. Lock and key model states that the active site of the enzyme is rigid and is not flexible. It acts as key and it attached to the substrate which is exactly matching or similar to the active site of this enzyme, and the substrate acts as lock. So this enzyme will break down the substrate and lock and key model will be followed. In this case, only the enzyme or substrate will bind if their active sites or their attachment sites are exactly alike. So this is very specific model. While induced fit model states that the active site of the enzyme is flexible, it changes and accommodates or induced changes according to the substrate shape in order the enzymes may bind to the substrate. So this is flexible model, and the active sites of the enzymes are continuously undergoing the conformational changes in order to fit the substrate in these active sites. So this is a flexible model. But here the example, urease enzyme convert urea into ammonia. The urea is converted into ammonia by urease enzyme. So this is basically a specific enzyme, the urease, which can only bind to the urea. It has only a single substrate, the urea. So it is very specific enzyme and it follows the lock and key model, the specific model, in which the enzyme will bind only to the substrate which is exactly alike to their active sites. So this will follow the lock and key model, in which the active site is non-flexible and it is rigid and it only binds to its own substrate. The urease enzyme will only bind to the urea. So this is following the lock and key model, not induced fit model. Next question is, enzymes and coenzymes belong to dash category of the biological molecules. Given options are same, different, remarkable, or not comparable. As we stated in this lecture, that enzymes are all proteins in nature except the ribozymes, and coenzymes, these belong to the category of nucleic acids. These are basically made up of dinucleotide units. The coenzymes NAD, FAD, NADP, or we can say the nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate, and flavin adenine dinucleotide. So these are the three coenzymes which are used in the body: NAD, NADP, and FAD. These are the coenzymes which belong to the dinucleotide or nucleic acids, while enzymes, these are proteins. So enzymes are proteins and coenzymes are the nucleic acids. So both of these belong to different categories of the biological molecules. So option B is correct, that the enzymes and coenzymes, these two belong to the different categories of the biological molecules. Next question is, malonate is a dash kind of inhibitor. Given options are competitive, non-competitive, reversible, and irreversible. So before going to solve these MCQs, we should know about the malonate. So malonate is a kind of inhibitor as it is stated in the question. In case of Krebs cycle, succinate is converted into fumarate in the Krebs cycle by succinate dehydrogenase enzyme. Succinate dehydrogenase. So this is the enzyme. And the inhibitor malonate binds to this enzyme. Malonate inhibitor, it always binds to this succinate dehydrogenase enzyme, but this binding is conditional. Malonate is a competitive inhibitor and it is in competition with the substrate succinate. So succinate and malonate inhibitor, these both are in competition. Succinate is a substrate and malonate is an inhibitor, and succinate dehydrogenase, it is the enzyme. Concentration of succinate substrate is decreased in the cell, then this malonate will bind to this succinate dehydrogenase enzyme in order to prevent breakage or utilization of this succinate dehydrogenase enzyme. And whenever the concentration of succinate is achieved, malonate will be detached from this enzyme and succinate will be converted into fumarate. So basically substrate succinate and malonate inhibitor, these are in competition. So this is a competitive inhibitor. The option C is also correct, because malonate is a reversible inhibitor. When the concentration of the succinate is achieved, malonate leaves the enzyme. But here reversible and competitive, both options are correct, and we have to choose the most appropriate, the most specific one. So competitive is the most specific, most suitable option. So this was all about the MCQs pertaining to the enzymes chapter. These MCQs will make you learn many concepts of the enzyme and will help you in preparing for your medical colleges admission test. Thank you.